So just as background as to why we got into this and why we've moved so deeply into the biofuels area, this is a graph that shows the projected use of energy over the next, well, the past and going forward. So the end point on this is actually going out to 2035. And if you do some quick math in your head, you can very easily see what the nature of some of the problems are. So this is in quads per year, and 2035 is projected to be around 720 quads. But if we look at actually the amount, and so one thing to recognize, this is total energy, but from petroleum, that is about 35 to 40% of the total energy used in the world is petroleum, and that varies. And with a lot of nuclear power plants coming offline, that number actually will probably go up. It's expected, however, that the amount of energy used will not decrease in any substantial way. So, so if you consider that this is about a third of, or so of this is actually in the form of petroleum, that gets us out in the order of 2,500. And then if you look at the total number of barrels of oil which are in the oil reserves of the world, what you see is actually that number is about 7,500. And if you start to do the math as to what percentage of the total amount of oil that we have in reserves are used annually, projected out around 1935, you rapidly come to the conclusion that we're going to pretty quickly have the amount of oil, and within your lifetime, the amount of oil will definitely be brief. And again, this is, if you do the math, it's a kind of a scary number. By the way, you should be scared because these oil reserves are actually pretty accurate. And so these numbers are really fairly well known. I know there are changing technologies in terms of how we can get oil out of the ground these days, but they're not changing that much. And these reserves really do reflect the amount of oil that we have. So where we have our interest is, it does revolve around liquid fuels, but this graph sort of illustrates the different types of fuels. So this is actually gasoline, the dark blue, and this lighter blue actually represents highway diesel. And actually included in that, in addition, is fuel for jets, jet fuel. And we've become interested mostly in this part of this, this equation in terms of the diesel. And the reason for that is it's a long-term solution that we don't really have, and we are not really well versed in terms of how we're going to solve that. And this is a bigger problem than gasoline. And it's a bigger problem, even though that's a smaller part of the pie, because I think most people would say, unless they have a remarkable crystal ball that I'm not aware exists, we really don't know in 20 to 30 years what the nature of highway fuel is going to be. We already see that there's a very large switch towards hybrid cars, electric cars, and it would not be unreasonable to project that in 20 or 30 years that maybe the amount of gasoline will be decreased just due to technology advances, not to increase gasoline, but rather just technology will move that away. But in the context of jet fuel and diesel, there's virtually no projections of that going away. There's no projections that we're going to have battery-operated jets or long-haul trucks. So we actually have, it's a situation where we don't really know how we could replace that. And in fact, if you look to see what research is being done, that's, say, supported by the Department of Energy for the replacement of biodiesel, the answer is virtually none. And the reason is because there isn't a good substitute. So when it comes to diesel, this is something that in the long haul we really would like to be able to make sustainable. We'd like to make it sustainable even though it's a smaller part, not only of course because of the fact that petroleum reserves are going to be going down, but also just from the standpoint of climate change. So all of the work that I'm going to talk to you today about really has as its driver the idea of producing a biodiesel or oil, in other words. Whoops, wrong way. So in order to do that, what are the possibilities? And as soon as you start thinking about the possibilities for substituting a renewable form of diesel, you run into a controversy that is actually as much a political controversy as anything, but it's a very real controversy, and that is the argument of food versus fuel. As the price of agricultural goods goes up, inevitably the argument is made that that's because it's going towards fuel. I think, by the way, that's a false argument. I think the agricultural economists are not at all in agreement that that's what's going on. But it doesn't make any difference, actually, what's going on. The perception is that that's what's going on, and perception is everything in this context. So whatever we want to do, we're going to need to think about coming up with alternative feedstocks for producing things like biodiesel. I think, by the way, that's also going to be true for gasoline, but because this, again, this controversy won't go away. So when we think about biodiesel, what are the potential crops that we could have, and how do we even want to think about them? And I think there are several possibilities or things that we need to consider. One of them is, whatever we do, if we want to have a crop that's going to be used primarily for industrial applications, it should not be a food. So corn and soybeans should just sort of be taken off the list of things that might be used for producing biodiesel, or for that matter, I would say almost any fuel. And the other thing that would be really useful is that whatever that crop is, it doesn't grow when or where soybean and corn grows. So it's not good enough to just simply substitute the crop. You can't take away any of the corn or soybean, and that means really it needs to be growing at either a different time of the year, which is, I think, quite a reasonable thing, or in a different place. And there's a lot of good candidates for growing in an alternative place. In fact, the Great Plains, which is not really well populated in terms of agriculture, at least dense agricultural practices as we have here in places like Missouri, Illinois, Iowa. If we can come up with crops that grow well in those areas, that would be a really good alternative, and would not greatly decrease the amount of food that's produced. And so in my lab and in the grants that we're operating on, we actually have come up with 3 that we have focused on. These are not the only ones that are out there, but there are 3 that we have focused on. One is algae, and I think for this group, I don't really need to go into this very far. I think algae is sort of an obvious and acceptable alternative. There's a lot of technical issues associated with algae, but it's still, I think, a very viable candidate. The one I'm going to talk to you mostly about today is Camelina sativa, or Camelina. This is a relatively new crop, and I'll expand on that. Another one is Castor, Ricinus communis. This is one that gets really very little press, and I'll take just one slide to talk to you about why Castor is a candidate. Castor has a huge number of pros associated with it. So it grows on very marginal land. Marginal land meaning, it's not quite desert, but very little water. It grows, and it's often the case where there's very little water. Those soils are saline. Very few plants will grow on saline salt. Castor grows wonderfully on saline salt. Its oil content in a seed is 60%. It's amongst the highest of any plant that exists. And the oil yield per acre is amongst the highest of any plant in the world. Palm is the best. But Castor ranks up there with it. So why isn't it an oilseed crop? And the cons are one huge one. It's called ricin. Ricin is a poison that's amongst the most potent poisons in the world. The LD50 is 22, whoops, didn't come out there. It's 22 micrograms for an LD50 will kill an adult human. Now I have to say that that's not by taking it orally. If you inhale it, or by injection, 22 micrograms. There are very, very few poisons that exist that are that potent. And the sole source of ricin in the world is castor. It's found in the seeds, seed coat, not in the main part of the seed, but in the seed coat. It's found in the leaves, the stems, virtually all over. So farming with ricin is potentially hazardous. And in this country, it's probably not going to happen as long as there's ricin in there. And of course, today, so ricin, by the way, is a protein. And so the obvious way to get rid of ricin is just to do an RNAi knockout. And there's no doubt, by the way, that could be done. It can't be done by breeding, by the way, because there are 8 copies of this gene in castor. And so they've got it down to about 30% of what it normally is, but that's not nearly good enough. You'd really have to knock it out. But the gene sequences are known, really not a problem. So why don't we do that? And that's the second problem. Genetic engineering capability is essentially undeveloped. And it's not due to lack of effort. I can tell you, we've put 3 years of it in my lab. But it is a euphorbs, and euphorbs are notoriously difficult to genetically engineer. And I think most people would admit that this is the most difficult nut anybody's tried to crack yet in the genetic engineering world. Talk to tissue culture people, they'll say, if you beat on something hard enough, you'll always get it to be genetically engineered. And I think we may have the example where this may not be true, but we're still working on it. But you can imagine, though, the day that we genetically engineer Castor, the first thing that will happen is we'll knock out ricin, and then all of these things will be true, and I think we'll have a crop. So we're essentially one successful experiment away from developing a new crop. And so this is the way I think is a good way to think about some of these things. Some of these problems are difficult problems, but they're a single problem that once resolved, we could then plant this in literally millions of acres. If you've ever flown from Dallas to San Diego, you spend a significant amount of time flying over land that nothing grows. That's called Southwest Texas. And Castor grows very well there, and that's been demonstrated quite clearly. So there's hope in terms of some of these crops. I'm going to take a few slides to talk to you about the programs that we're running that are supported by DOE. One of them is CABS, the Center for Advanced Biofuel Systems. The people who are part of this are listed here. Most of them are at the Danforth Center, although Ed Cahoon and Dick Sayre were originally at the Danforth Center, and they've subsequently moved on to other institutions, but they're still part of CABS. CABS is heavily focused on Camelina and algae. The rationale, this is as many of the EFRC programs are, is really just a wonderful grant to have. It really asks the question, how similar are algae and plants with respect to producing oil? And at the fundamental levels. We know a lot about how oils are produced in seeds. We don't actually really know that much how they're produced in algae. And so the question is, what information transfer can we use? And in the process, we are really expanding our knowledge of the seeds as well. So this is sort of a cartoon to say where we're focused. So PARC, which you're all familiar with, is focused up here in terms of light harvesting complexes. And we take that energy and we study everything else, mostly focusing on oil. Actually, studies dealing with starch and other components are mostly from the standpoint of knocking them out to see if we can, in fact, move more carbon up into oil. But we're doing some work with the light reactions, certainly with the Calvin cycle, and then metabolism to oil. And this is I'll illustrate this further in a later slide, but there's a lot of complex reactions, metabolism that's occurring here. So CABS is really a focus on metabolism more than anything else. The two systems that I said we're working with are microalgae. I will say briefly that microalgae are significant because you can get as much as 40 to 50% of the biomass as lipid, and actually you can harvest 100% of the biomass, which is difficult to do with plants. And of course it has its advantage in that it can be grown 24/7. It grows, its doubling time is much greater than that of a plant. There are many advantages to algae. So the questions that we're asking is, okay, so just how does this organism produce oil? What's the basic metabolism? I would say, and I'm not going to talk about the results from CABS, but I think one thing I will say that when we started this program 3.5 or 4 years ago now, almost 4 years ago, we thought that they were going to actually be quite similar because of publications that occurred in the 60s and early 70s suggested that there wasn't much difference between algae and seeds. I can tell you today that we know that's absolutely not true. That there not only are differences, but the differences are large, to the point that I think we're back to ground zero with algae. in terms of trying to understand the metabolism. There's very little transfer. And in fact, maybe algae can teach us something about plants that we didn't know. The algae that we have been working with as a model is Chlamydomonas, and specifically, we've been working with star 6. This is a starchless mutant of Chlamydomonas that produces large amounts of oil. It was actually isolated here in Ursa Goodenoug's lab. And it has all the advantages of being a large oil producer, and it's Chlamydomonas. So in terms of algae, this is really a big deal of having something that we actually have some good genetic tools to work with. For virtually all the other algae, that's simply not true. And so they're very difficult to work with as organisms. We're trying to catch up with that. The field is trying to catch up with that, but it still has a long way to go. So I think there's no one who really believes Chlamydomonas has any place as an industrial algae in the future, but right now it's the very best learning tool we have. The other plant that we're interested in, Camelina, and we're interested in specifically from the standpoint of asking, can it really be an industrial crop? And the first thing one might ask is, why are we interested in Camelina? And there's a number of huge advantages that are associated with Camelina that simply don't exist for any other plant other than Arabidopsis. And first of all, while it's not Arabidopsis, it's amongst the most closely related of plants to Arabidopsis. Its genome, on average, its genes are frequently 90 to 95% identical at the nucleotide level to Arabidopsis. So that's about as closely related as you're going to get. Genetic engineering is just the same as it, or almost the same as it is with Arabidopsis. So it uses the flower dip method of transformation. And what that allows you to do is get a lot of transformants really, really easily. For those of you that are not aware of how this works with plants, all other plants, I know of no exceptions to this, other than Arabidopsis and Camelina, require going through a tissue culture procedure in order to get a transgenic plant. And that, from the day you hand over a construct to somebody involved with tissue culture, you're going to be waiting anywhere from 12 to 15 months before you get your first seeds that you can start doing some work with. And you're going to pay a lot of money, by the way, in between. So it's an expensive, costly procedure. And if you get 10 plants, that's going to be a happy day for you. When we work with Camelina, on average, we'll get 45 to 50, frequently as many as 85 transgenic seeds as a transformation. And when you're playing with what are frequently quantitative traits, having a large number of transgenic plants is different independent lines is extremely important to the success of the research. A lot of the genes that you get in simply don't work as well as they do in other plants. And what you want to do is study the ones where they're working really well. And so this requires a lot of transgenic lines. It goes about 3 months from seed to seed. That's not as fast as Arabidopsis, but it does allow us to get almost 4 crops a year in the greenhouse. So that's quite good. The seed size is 1 milligram. And that's small, but it's not too small. And the important thing is normal farm machinery can use Camelina. So if we're thinking about an industrial crop, what does that mean in the context of acreage? And so to give you a context, last year soybean was grown at about 70 million acres. Corn's about 90 million acres. So when you're thinking about an industrial crop, you're talking in the tens of millions of acres. And that means this is not going to be grown by hand. It's not going to be grown in your backyard. It means it's going to have to be fit into the main agricultural infrastructure. And that infrastructure starts all the way from the farmer, from planting it, to harvesting it, to getting it to grain elevators. All of that infrastructure needs to be in place if you're going to be dealing with something in the tens of millions of acres. So we were only interested in plants that it could fulfill that limitation. Today it has very, very limited use as a food crop. It is harvested probably a few thousand acres for oil. Again, when you think in the tens of millions of acres, a few thousand acres is nothing. And while there were really no genetic resources available until recently, at the Danforth Center, we've actually put a major effort into getting those genetic resources. So we now have the genome sequence. We don't have the first draft yet of that, but we at least have all the data. We have done extensive RNA-seq analysis with this. And so things are moving along, and I think within probably the next 6 to 12 months, we'll have many, many of the genetic resources that are really necessary to push this plant. So a lot of positive things to be said about Camelina these days. The other grant that we have from DOE, which we got relatively recently, less than a year ago, is one we call the Center for Enhanced Camelina Oil. This is an ARPA-E grant, which is $5.5 million over 3 years. When in the plant world, a 3-year grant is about the most minimal grant you can possibly get because it does take time to grow them and harvest. This is a very short grant. It's almost a perfect complement to CABS in that CABS is to undermine the fundamental research or a fundamental basis for how oil is synthesized, either in Camelina or in Chlamydomonas. This is totally focused on Camelina and it's completely translational. All they want is more oil and they want higher yield. And so what we're doing is taking many of the genes we've already been studying in CABS and combining them to see how that would work. Because we know how these work individually, we have no idea how they work, if they work synergistically or possibly antagonistically. So this grant is to do a lot of genetic engineering with a lot of genes. And so what we're intending to do and what we actually have largely accomplished is to intensively genetically engineer using 12 to 16 genes in combination in the same plant. We haven't got 12 or 16 in one plant yet, but we have one set of plants that has 8. We have another set of plants that has 4. Ultimately, these will be either crossed or we'll add more plasmids to it. So, so basically, we're attacking the plant both at the leaf level and as well as at the seed level. And in the process, we're doing a lot of phenotypic analysis to make sure that what we're doing is working well. So we're doing both molecular as well as whole plant physiology to make sure that when you put something together and you if we get a lot of oil or get what appears to be a lot of a good yield in a greenhouse, that actually this is going to translate out into a field. Because this is, the ARPA-E grants really are for looking at projects that are going to ultimately get into the field. So it's to take that potentially applied research and apply it. And that's what this is for. Which actually, by the way, I might add, it's the first grant I've ever had in my career, which has been a long career now, where I've actually been asked to actually translate the work that we've done. I think in the many grants that I've had, they've all been to do basic research. And so this is it's kind of refreshing to actually be able to see if something will work in the field. One of the things that allows us to do this, this work in a relatively short period of time is we have vectors now that will carry at least 3 cassettes. We actually have one that carries 4 cassettes. So it's 4 different genes independently controlled by different promoters and terminators. And in addition, we've been using something called DSRed. It's a fluorescent protein that is very easy to, it's very bright. When you shine a green light on it, the tissue lights up very brightly. Which is much brighter than GFP or any of the other fluorescent proteins you see. And so after you've done a transformation, you can simply shine the green light on it, and it's pretty obvious which ones are transgenic and which ones are not. And this, I can't overemphasize enough how important this is to a research like we're doing in CABS, rather in CECO, as well as you'll see another project that we're working with. Being able to look at this, so these are essentially T1 seeds that are that you can see when they're first transgenic, and so you know every red seed is a transgenic seed. Even though it may be heterozygous, you can still do, if you've got enough of them, you can actually do some research on them right away. So when we transform when we transform Camelina, about 6 to 8 weeks after that, we have our first set of seeds. If we've got 45 to 50 transformed seeds, it's pretty easy to just sacrifice half of them and do some research on them right away and plant the other ones so you can get homozygous lines and do further research on them. And that's frequently what we do. We do the first T1 seeds we'll actually do some experiments on. And again, if you're working with tissue culture, that's simply not an option. Those seeds are few and they're precious. And so you do no experiments on them, you plant them. So I'm going to diverge away from here. So this is basically, I've told you about some of the large projects that we're running from the Danforth Center dealing with trying to increase oil. I want to talk about another aspect of genetic engineering and oilseed technology that is near and dear to my heart. So as we think about petroleum and thinking about sustainability in the world, as you all know, most of the petroleum in this world is used for fuel, 90% of it. But the entire chemical industry, Dow Chemical, Bayer, BASF, the chemistry that they rely on comes from the same barrel of oil. It comes from petroleum. And so, in fact, that industry is very often referred to as the petrochemical industry. Virtually every plastic you've ever used or is around you has come from petroleum. Okay? So the question that we want, so there are two questions. If we're going to really truly have a sustainable world, what's going to happen to our plastics? They're probably not going to go away, but we'd prefer that they not come from oil. And in fact, Dow Chemical will tell you they almost certainly won't come from oil 30 or 40 years from now, because the history of the plastic industry is that plastics are cheap. And so the first thing that they want to do is keep their plastics cheap. And if petroleum becomes expensive, they want an alternative to petroleum. So we've been interested in how can we begin to think about how we can substitute petroleum with sustainable sources of carbon. And so my area of research is oil synthesis, fatty acid biosynthesis. And if you actually look across across nature and ask, so what kind of chemicals are out there that might be useful? It turns out almost everything that's in a barrel of oil you can find in nature. That's, after all, where it came from in the first place. And so when you think and you talk with people, the chemists at Dow Chemical, which I've spent actually a fair amount of time doing, they'll tell you that there's a number of functionalities that are really important. Hydroxy groups, epoxy groups, conjugated double bonds are all extremely useful chemistries for producing polymers of various sorts. And actually, all of these are quite common in nature. When I say common, not to say they're not found in soybeans or in corn, they're not actually found in our food at all. But they are found in plants that are around us. And in fact, I talked to you earlier about castor. 90% of the oil that's in castor is that hydroxy fatty acid. And this is actually one of the ones we study the most, because we know that castor is quite capable of producing this in very high quantities. And so we're interested in particular in this one because we know the machinery is well tuned in castor to do that. This is what's found in tung oil. It's what causes paints to polymerize. And the epoxides or epoxy plastics and paints are really very common. So many of the chemicals that we might want to think about getting are out there in nature. Invariably, they're found in places that are not useful from an agricultural standpoint. Well, this is probably an exception, but these, where you often find them, are in seeds that are in plants that grow in ways that are really not amenable. In fact, this is found in the tung tree. It's tung oil. It's the basis for tung oil. And seeds from trees are not compatible with agriculture. And in fact, one of the fatty acids that I dealt with had a hydroxy group here, right at the very end. And it grew in a tree that was called the Camelina tree, which grows on the eastern shore of Australia quite sparsely. And in fact, every tree has just a few seeds on it. So getting the desirable fatty acid from one of these sources is really not in any way possible. So the idea, of course, is to harvest these plants, get the genes out of something like castor or the seed of a tung tree, and put them into a plant like Camelina that might produce large quantities. And I'd say 25 years ago, this was viewed as low-hanging fruit. This was something that we were going to be able to knock off in just a few years. People were thinking about forming companies. We were going to get rich. It didn't happen. I'm here talking to you. So I might not, if I was rich, I'd be retired in the Caribbean. So as we think about this, one of the things that's useful to think about is how the petroleum industry today works, because ultimately we want to replace this industry. But it'd be useful to know how the industry works. And so if you've ever been to a refinery or seen pictures of a refinery, you know there are these massive structures. And what you might not realize is within that refinery, they're not just producing fuel, but virtually all the chemical feedstocks that goes to places like Dow Chemical are coming out of that very same building. It's 100% integrated into the industry, so that the barrel of oil going in is completely segregated into the various components that it's going to go to in that building. So it's a highly integrated industry. In fact, the largest facility that Dow Chemical has is right next to one of the largest refineries in Texas. So they don't even transport it by truck or train. They just pipe it right into their facility. So that's about integration at an extreme. So what we'd like to do is think about you treating plants as our source of oil, and in that process, think about producing plants in which all the biomass is used, and then out of those seeds, at least part of it is going to be used for producing bioproducts. And so our dream is to actually think about plants in this kind of a context. But when we do that, and we think about, especially think about producing oil, how am I doing on time? I'm going a little bit slowly here. When we think about producing oil, this is actually a fairly complex process. So this cartoon is sort of illustrating two very big buckets of metabolism that we have to think about when we produce oil. This is a large plastid, to say the least, or chloroplast. And the important thing to know is that for oil synthesis, the fatty acid, the monomer component of triacylglycerol, is entirely synthesized in the plastid, or exclusively synthesized in the plastid. But all the oil that forms triacylglycerols ends up as triacylglycerol in an oil body, and all of that is occurring out here on the ER. And so there are two aspects of this. The assembly of the oil is separate from the synthesis of the oil, number one. But secondly, virtually this entire process that's occurring on the ER is membrane-bound enzymes. And if you know very much about biochemistry, you know the least understood enzymes that we have are membrane-bound enzymes. And so how this process actually works in the ER is really not well understood. However, I would say 25 years ago, we actually thought we understood it. Today, we've been humbled. And so here's where we are today in terms of the number of enzymes that are involved. And I'll take just a minute or two just to try and illustrate this complexity. So if you start with glycerol 3-phosphate and you add acyl groups to it, you eventually can end up with TAG. And a number of years ago, we thought this was actually what it was. It was about a 4-step process to produce TAG. What we now know is actually when you get down to here, you produce this diacylglycerol, and this virtually 100% goes over and forms phosphatidylcholine. And phosphatidylcholine gets modified. In this case, we're showing it so it's producing a hydroxy fatty acid, and then it gets cycled back. And it can cycle back by reversal of this process. It can cycle back by cleavage and go up and re-enter the process. It can cycle back by using an enzyme that's referred to as a PDAT. And in fact, there's recently been another enzyme that is believed to be different from PDAT that may also be involved in this process. So what we know is you have to go here, and then you have to come back by what we know are candidate enzymes, but in point of fact, we don't know what the process is. And the nightmare is we think all three possibilities are actually always active. And we don't know what regulates the process at all. And if you analyze oilseeds from plants, what you find is the composition of that tag, as well as the composition of the PC, are usually very different, number one, and they're absolutely fixed. So what that tells you is it's tightly regulated. If nothing else, you'd have to say it's tightly regulated. But what that regulation is, we don't know. But it keeps us busy. So because of that, how well have we done about producing unusual oils in seeds? So these are the percentages that you can frequently find in a native plant. So I was telling you earlier about ricinoleic acid. It's 90% in castor. If you produce transgenic plants, this is about what you get. The exception of medium chain fatty acids, they're all less than 25%. These are the absolute best numbers that are available. The norm actually is about 1%. So there are many, many examples where there are different ones, where it's 20%. We have actually now got this one up to be about 30%. That's really a world record for everything but medium chain fatty acids. But it's nothing approaching the 90%. And what I can tell you is the reason that we can't get it up higher is we don't know enough. We simply don't know enough about the metabolism that's associated with these processes. So what is the future of this? Well, I think the future is going to be based on really trying to understand much more about out the basic metabolism that's associated with this process, and trying to figure out what genes are contributing. And furthermore, are there things missing? And that is actually the one we really lose sleep about, because we've discovered within the last few years 2 new enzymes. So that kind of gives one pause to say, well, how many more are we going to discover? And so it may be that we're not even playing with a full deck when it comes to trying to assemble this process. And the other thing is there anything special about membrane organization that we're missing in transgenic plants? And I just want to dwell upon this because this is something, let me skip over that we think quite a bit about in my lab. And that is that when we think about targeting genes to the ER, we know how to do that, but actually we only know how to do it in the broadest of ways. So I could target a person to come into this room, but it's another thing to target them to a particular seat. And we only know how to target people to a room, but not to a seat. And the question is, how is the ER organized? And in fact, if just looking under a microscope, you can tell that it is organized. And there are various sections of the ER that have various contact points and other points. And there's this part of the smooth ER that produces oil bodies. So the question that we've been trying to ask is, where those oil bodies are produced, is that where all the enzymes are? Because actually, the biophysics suggests that diffusion through the lipid bilayer would work pretty well as well. So it doesn't have to be there. But in point of fact, if you ask, what's the evidence that they are there or they're not there, there is no evidence. It's a belief, a conjecture. But there's no scientific evidence that these things are really right next to each other. And so we've been trying to take both a proteomics as well as a yeast-2 hybrid approach to try and analyze that. What we're finding is there are a lot of associations in a native system. Now the question is, if you're trying to make a transgenic plant, do they assemble properly? And the answer is we have no idea as to whether or not that's the case. But that's the nature of the field today, is that there are a lot of questions. And so one of the things that I can leave you with this message is 25 years of doing research trying to do this has taught us that biology and biochemistry and metabolism are at least an order of magnitude more complex than we ever imagined. Probably at least an order of magnitude more complex than any textbook would ever lead you to believe. And so you have a fruitful career ahead of you if you're young, because we're definitely not going to solve it in our lifetime. Let me see. I have a few minutes. Let me quickly talk about one other project we have, which is a complete digression, but it's a story I love to tell. And it deals with producing another a renewable resource, a biomaterial that is not an oil, but is called a polyhydroxyalkanoate. This is a project that we got involved with a company in Massachusetts called Metabolics, who produced this biodegradable plastic in bacteria. And they're asking the question, can we do this in plants? And so the process for producing a polyhydroxyalkanoate is really quite straightforward. It's just 3 genes that you can get out of certain types of bacteria. And they produce essentially a polymer which has some really nice features to it. It's essentially a relatively hard plastic. The types of plastic forks and spoons and knives that we've all thrown away are essentially that kind of a plastic, that kind of a texture. You can make Gulf teas off of them, anything like that. What's nice about them is they're completely biodegradable. So you go to a landfill and about 30 days later, they're gone. And when they biodegrade to a molecule that actually is running through you and I right now and does every minute of our lives. These are just very small 4-carbon units that are part of our normal metabolism. And so they're as non-toxic as non-toxic could be. In fact, they're sufficiently non-toxic that if you broke them down and took these C4 molecules, you could eat them and as a replacement for sugar. It doesn't taste as good as sugar, but in point of fact, it metabolizes essentially the same as sugar. So a great product from the standpoint of sustainability as well as preserving the environment. So what our idea was, so this is the starting product, or the starting material is acetyl-CoA. And the idea was to produce these in plastids where we know there's a lot of acetyl-CoA involved with fatty acid metabolism. And so This was something we didn't know how this is going to work out because acetyl-CoA is the central molecule in metabolism. If you look at the sigma metabolic map and you look for one that has the most arrows coming in and out of it, it's acetyl-CoA. So what we're trying to do is divert acetyl-CoA into this new molecule. And so from the standpoint of metabolism, it was an interesting question as to how we could do this. Because unlike with bacteria, it's now going to be in the context of a multi-organellar cell. So we generated a plasmid. It's very similar to the one I showed earlier. Once again, it has the DSRed marker in it, which is really very important for reasons that will become obvious. And our very first experiment, we had a lot of transgenic seeds, sacrificed a few of them right away, and this is our product right here. This is the, this is the PHB, this is the internal standard, but we had a nice peak of PHB, completely missing from the wild type. Our target was about 8% PHB. If we could get 8%, it was going to be an economical project that we could make money on, or metabolics could make money on. I was in there for the fun. And so we got actually from anywhere from 0 up to 15%. So we were related of that. So the question was, how well would these seeds, how viable would these seeds be? And the answer was not very. So as it turns out, these are some of the starting ones, and I'm having a hard time seeing it from this angle. That's another one that's aborted. But essentially, all the other plants that were wild type were already plants. These were kind of stuck at the cotyledon stage. Many seeds didn't even come out of the ground. So, and these are, again, some of the transgenic plants. This is a wild type plant. So they're really very stunted. Eventually, by the way, once they got to about this stage, they would catch up. They would be fine. So the problem was actually the seed itself was not viable. We went back and we analyzed some of our seeds to see, all right, if we take a group that's 10% or 5% or 2%, which are the ones, where's about the cutoff point? It turned out nothing above 5% was viable. They would just not even come out of the ground. The ones that were stunted were in the 2 to 5% range. So it was really at a pretty early stage that these things are a pretty small amount of PHB that these things quit. And in fact, here's some of the seeds. This is a wild type seed, and you really don't have to worry about the details, but you can sort of see how this thing is distributed. This is a 2% PHB. This, by the way, is the PHB. And almost all the organelles look a little strange relative to these. And this is the 5%. And it's even worse. And I'm going to blow up this one. This is the 5%. And this is PHB inside a vacuole. And so what's happening is that plastid is undergoing autophagy, which almost never happens in nature. So this is what you call a royally messed up seed. And that it's not viable is completely reasonable. After we saw these pictures, it was pretty reasonable. So is this a failed project or not? And so the answer is not yet. We haven't succeeded yet, but we actually know where we want to go. So now we know what the problem is. The problem is that if you produce a lot of plastic, a lot of PHA or PHB in seeds, you generate a seed that's not viable. But 99% of the time, you actually don't care about that, because that's going to the grain elevator to be harvested. You didn't intend to replant it. You actually want to grind it up. So the ones that have the 15% are just fine. The problem is the ones that have 15% won't give you another generation. And so what we're working on actually is to change our strategy for how we go about doing this. So if we can get at least 15%, we are really in great shape. That's a wonderful product for us to have. The problem is we have to have some of the time where we don't get any. And so what we're working to do is actually develop a strategy where we can regulate PHB production. And the preferable situation would be that the default is the seed produces PHB. and that we have a way of spraying something on it so it doesn't produce PHB. So we would like to get some way of suppressing the synthesis of the genes, or the production of the genes, for producing PHB. We've actually not succeeded at that. Our first couple of strategies didn't work all that well. And I can tell you the reason for that is most of the strategies that are available depend on metabolism that's been developed and understood in the context of a leaf. And so one hopes that it's going to work in the seed as well. And so far, it hasn't worked as well. And many times, it's delivering an inducer. It's much easier to spray it on a leaf. It gets absorbed by the leaf, and it works beautifully. For a seed, you can't spray it on the seed. You have to spray it on the plant, and it then has to be transported to the seed. And so it's a transport problem. And so we're now trying to think of strategies to either change the transport issue or change the molecule. But conceptually, it's a pretty reasonable approach. And in the meantime, we're having an awful lot of fun working on a project that I think potentially has this type of an application. We're learning a lot of biology. For example, the idea that some of these inducible promoters wouldn't work in the seed was not in the literature before. There was no reports of that. So we've learned something quite useful from that standpoint, because there are many applications we might have for trying to induce genes in a seed. And we now know that's not nearly as easy as we thought it might be. And so I think with that, yeah, my last slide of this kind of messed up too. And Claire got wiped out entirely. But in any case, these are the people in my lab who have worked on various projects. Some of them have moved on. Jonathan Markham is now has a position at UNL. Joel is working for Dow AgroSciences, but a number of these people are still in the lab. Funding came from a variety of resources. The PHB project actually was funded by the state of Missouri. And then DOE has been by far our major funder in the last few years. And these are a number of my collaborators who are involved in these various projects. And with that, I'll be happy to take questions.
Questions for Jan? Maybe I'll start off.
Yeah.
The Camelina is a really interesting potential crop. What kind of harvest index do you get out of it? In other words, how much seed do you get out compared to the total biomass? Is it something that is going to, can you engineer that or increase that, you know, like has happened with corn and soybeans and so on?
That's a really good question, and it's actually one of the reasons that I'm optimistic about Camelina. So the history of Camelina is that it was grown, I don't know what its origin is, right? I believe it was Eastern Europe, but regardless of that, the place where it really impacted in agriculture was in Northern Europe. Essentially, every place today that rapeseed grows, which in Canada is canola, every place that rapeseed grows in Europe used to be Camelina. But about 100 or 150 years ago, they recognized the yields on rapeseed were higher than Camelina. And Camelina virtually disappeared from agriculture, certainly in Europe, but actually across the world. So, and that was, again, 100 to 150 years ago. So breeding stopped then on Camelina, and rapeseed really started. And so rapeseed has now gone through many years of modern breeding programs. Its oil has been increased from what used to be in the high 30s. It's now at 50%. That's actually happened relatively recently in the last decade with very modern breeding techniques. And its yields have increased tremendously. Camelina has gone through none of that. It had no breeding whatsoever in the context of what we think of as breeding. So that gives one pause. The other thing is that the accession lines that are available have not been very starting. So the one that we're using is 30% oil, way below what rapeseed is. But we've just now examined about 60 or 70 different accessions, and we found one that's over 40. We were using because that's the original publication on genetic engineering of Camelina used a line that's called Sunison. And I really didn't want to be experimenting with different lines, so we use Sunison, and it works wonderfully. But now we've identified a second line that's much higher oil, and we've demonstrated that it's easily transformable. Not as good as Sunison, But when you're as good as Camelina is for transforming, it's more than good enough. It's really excellent. So that's not even breeding. That's just testing a few lines. So the anticipation is that places like Montana State and probably Washington State, that Camelina will start to enter into a breeding program. And with all the genetic engineering we want to do, to be honest, I'm more optimistic we'll get more out of breeding than we will out of genetic engineering. But the idea, of course, would be to combine the two and see how we can go. So right now, in terms of yield, the question that's sort of in the other side of your question is, why would any farmer plant Camelina? And there's only one reason for them to do that, is if they're going to make more money than if they were planting something else. And so today, they would not. They would not, at least not in a place like Missouri or Iowa. But in Nebraska, in western Nebraska, out on the Great Plains, or in Montana, it's close. And the reason it's close is because Camelina requires less fertilizer, less inputs. And so you can you take a yield hit with Camelina, but you also save in terms of the input. So right now, it's close to break even. It's still not being planted in large quantities. And in fact, I think last year was like 50,000 acres, which again, in the tens of millions, this is not anything that's important. But largely, so the USDA was trying to promote Camelina to be grown, and they really weren't taken up on it by the farmers. And the reason is they haven't grown it. So this is another part of agriculture that people forget, that if you're going to grow things in the tens of millions of acres, I hate to keep coming back to that, but that's the fact that we need to address, that it's going to start with a farmer. And farmers will plant exactly what they want to plant, and nothing else. And so unless you can convince them that this is something that you're going to get more money for, that you will be successful, that it's worth your while to do this in the long run, they'll plant what they've normally planted because they already know they can make a good profit from that. So we've got to get Camelina to the point that it's profitable and they see the profit. And that's really the big deal. So this is a really complicated issue. It's very different from the castor issue because Camelina will compete with other plants that farmers, it'll go into normal fields. And we need to outcompete whatever else they're growing, alfalfa or whatever. Castor's in a completely different situation because we can put that where nothing else is. So that's an absolute win-win situation. But Camelina is not there yet.
So its water requirements
are less,
similar to?
It's actually less than, the inputs and water are less than rapeseed. It will not grow I don't want to misrepresent this, it won't grow in arid soil. But the Great Plains gets much less rain than Iowa and Illinois. And Camelina grows fine there. Actually, that's where most of it is being grown today, is out in regions like that. North Dakota is another region where it's clearly growing well, but it's still not really profitable. And again, it's convincing the farmer that this is going to be profitable. So USDA tried using subsidies in the Northwest last year or 2 years ago, and it wasn't successful for that.
Other questions? Do we have any online questions? Will Dan be the one that would know that? Yeah, go ahead and I'll find out.
Well, I remember being in the room.
Remember to repeat it.
Texas, I sat down downtown talking about the price of the Castor. Clearly that hasn't happened. But I remember it was brought up in the room of mine after that was invested in making quite heavily. You meant to make the price of Castor was that feel viable like that. Have any field trial or anything like that been done on solar Oilseed plants yet?
Yeah, okay, so the question is, with respect to castor, if we knocked out ricin, would we have a plant that is less hardy or less agronomically favorable? And of course, we don't know the answer to that question. I'm not sure I would, by the way, agree that Castor is heavily invested in this particular protein. It's not present in large quantities in Castor. Thank goodness, it'd wipe us out. But so to give you a sense of, it's found. In fact, I don't think its function is known within the plant. The only way that one would suggest that it's heavily invested is there are 8 copies of the gene, but the quantities are not large that are produced in it. So, for example, it would take if you ingest ricin, because it now goes down into the gut, it's a completely different physiology in terms of how it affects you. So it takes 100 times more, actually, ricin to, it probably won't even kill you, but it'll get you extremely sick. So that's about 20 or 30 seeds. And virtually all of that's in the seed coat, which is actually going to be quite accessible in the gut to getting that ricin out. So for that many milligrams, for seeds are actually quite large. I would say that's not a large quantity. So for example, the quantities that are, it's not like a seed storage protein, where I would not even want to imagine the possibility of trying to get rid of a seed storage protein, because that's a heavy investment that seeds have. Ricin doesn't fulfill that category. And it's found throughout the plant. And so, again, seed storage proteins are only found within the seed. So I think ricin has a very different function in the castor. And I think, so as far as I know, the lowest percentage of castor that's known is 30%. And that's been through extensive breeding. I don't think it's gotten below that. And everybody who I think, people like me, say we're going to get it down to 0, are talking genetic engineering. And so we'll have to see. It's been a very slow slog, though, getting the genetic engineering to work. These things just die right off.
Questions? Yeah. Go ahead and ask at least one of them now. There are people online that I think would like to hear.
Yep. Oh, yes. So we've been, okay, so the question is, in the ARPA-E CECO grant, we had one of the targets was PS2, and it's the PS2 system. So what we're interested in doing there is seeing if, the observation is in algae, that if you decrease the amount of photosystem 2, you can increase the efficiency of photosynthesis. And so I can't really talk right now about that phenotype, but the idea is what would happen if you increase the photosynthetic efficiency within a leaf? So this is a really good question, because this is, I tend not to think so much about this particular part of the project, because the way the ARPA-E is working is we're working on essentially 2 sets of genes, and the sets are divided based on how much we know about the genes and how much their effect is. So we have what we consider our good bets, and we consider our high risk. And that particular project's in our high risk group. And so it's not combined with any of the other mainstream, or the main flow of genes that we're combining. It's being held separate because we've not tried this with a plant before. We've tried it with, as I say, in algae, and it seems to work quite well in algae. And so we're in the process of evaluating that. So the idea is to actually target chlorophyll A oxidase to decrease the amount of chlorophyll B, and see what that does. And at least I'm not aware. This is something that's actually being done in the Sayer lab right now. And that, I'm not even sure they have homozygous plants yet.
There's been a lot of that in clam
Right, that's exactly right. And that's the basis of the,
Right.
yeah, yeah, yeah. We've not,
Some of that's even part of PARC.
yeah, oh, is that right? Right. So the, in the cabs, the EFRC, we actually decided not to do that in Camelina. Even though actually, conceptually, that would have been the right place to do it. But we didn't have the resources to do it within the cabs grant. So we switched it over to CECO because for reasons which were surprising to us, DOE was quite interested in us doing what was really I mean, I'll be delighted if this works, but the rules obviously are very different in a plant than they are in an algae. And so, as a number of us have said, there's a lot of ways that this can go wrong. However, that wouldn't be a terribly big surprise. But if it succeeds, that's really great. And if you've got the resources to try the experiment.
Okay, any other questions?
There's a question back. The answer is I don't know, and I'll tell you. So the question was with respect to the genetic resources we're developing for Camelina. Are we also going to be looking for developing SNPs? And that has been just sort of a casual part of the conversation. I think it's going to have to wait to see how successful we are with with getting the genome, which I think is going to be a large effort. And then, so it's a hexaploid to begin with. So that's the whole problem right now. We would have the first draft if it wasn't a hexaploid. Exactly, exactly. So we actually have much farther along on getting the RNA-seq and microRNA analysis done. And I think that's going to come out actually before we have the genome sequence. But then whether or not we develop SNP resources, I think, is going to depend on other people's interests within the Danforth Center. So this is a collaboration between 4 labs at the Danforth Center. And this is sort of what we've decided to bite off right now. And I think we'll just have to wait and see. But it's a great idea, and it's something, so I wouldn't do that myself because I'm a biochemist. And as a biochemist, I just don't ever think along those lines. But I'm hoping that we'll develop enough at the Danforth Center that other people will say, hey, this is something that we'd like to do. Then you should come over and talk with us, because I think actually we would like to develop the Camelina community. It's a very small community right now in the scientific community. There's only, well, as of about 2 years ago, there were only like 4 labs in the world that were really working on Camelina. This is really exploding right now, and it's great. From my viewpoint, I don't even, there's so much to do that it's not even a matter of competition. It's just an overwhelming amount of work to do. So, so we'd love to have some other ideas coming in.
Okay, on that note, let's thank Jan for a really exciting talk.
Thank you.
Thank you for coming. Had a small but enthusiastic.
It's totally enjoyable. And I don't have to do anything there.